Hello everyone, we are from Group 3 and in this video we will be discussing the Shikimic acid pathway, which is an important biosynthetic pathway found in living organisms. And this is our group members. Okay, my name is Muhammad Shawal bin Nordin and I will introduce about this Shikimic acid pathway. The Shikimic acid pathway is a metabolic pathway used by plant, bacteria, fungi, and some microorganisms to produce aromatic amino acid, which is phenylalanine, tyrosine, and tryptophan. These amino acids are essential because they are required for protein synthesis and also serve as building blocks for many important biological compounds. Interestingly, humans and animals do not have this Shikimic pathway. This is why this aromatic amino acid must be obtained from our diet. Because this pathway is absent in human but present in plant and microorganisms, it has become an important target for herbicide and antibiotics. In this video, we aim to provide a clear explanation of the Shikimic pathway as an important biosynthetic route. We will discuss the key enzymes involved and explain how they catalyze each step of the pathway, supported by the relevant chemical structure and reaction pathway. This video also highlights the industrial and biomedical application of the Shikimic pathway and briefly introduce analytical or bioassay method related to the compound derived from this pathway. For the background theory, biosynthesis occurs through a metabolic pathway which allows cells to control product formation. This prevents unwanted side reaction and use energy efficiently. In this context, the Shikimic pathway plays a role in connecting primary metabolism to the biosynthesis of aromatic compounds. For point 2, the Shikimic pathway begins with intermediates from glycolysis and the pentose phosphate pathway. This simple molecule undergoes several enzyme-catalyzed reactions including condensation, reduction, dehydration, and phosphorylation, which leads to the formation of Shikimic. For point number 3, chorismate is produced as a key intermediate. Chorismate then serves as a precursor for the biosynthesis of the 3 amino acids, which are essential for protein synthesis, enzyme function, and various structural and regulatory roles in cell. For last point, the Shikimic pathway is biologically and chemically significant because it constructs aromatic rings, which provide stability and functional diversity in biological molecules. Since this pathway is absent in animals, it also serves as a selective biochemical target in agriculture, medicine, and biotechnology.
Hi everyone, I'm Hwei Xin. I will explain the key enzymes involved in the Shikimic acid pathway. Overall, the Shikimic acid pathway consists of 7 enzyme-catalyzed steps. These enzymes work one after another, starting from DAHP synthase and ending with chorismate synthase to guide the pathway in a stepwise manner. Although the pathway consists of 7 enzyme-catalyzed steps, I will focus on 3 key enzymes that are especially important for regulations and biological significance. Without further ado, let's start with the first key enzyme, which is DAHP synthase. DAHP synthase catalyzes the first committed step of the pathway. It combines phosphoenolpyruvate and D-erythrose 4-phosphate to form DAHP via an intracellular aldol reaction. Because this step controls the entries of metabolites into the Shikimic acid pathway, DAHP synthase plays an important regulatory role. Next, let's move on to the second key enzyme, which is Shikimic dehydrogenase. This enzyme converts 3-dehydroshikimic acid into shikimic acid using NADPH as a reducing cofactor. This step is significant because it leads directly to the formation of shikimic acid, which gives the pathway its name. The third key enzyme in this pathway is EPSP synthase. This enzyme catalyzes a late step in the pathway, leading towards chorismic acid formation. EPSP synthase is well known as the target of the herbicide glyphosate, and it is absent in humans, making it an important target in agriculture and antimicrobial research. Finally, the pathway concludes with the formation of chorismic acid, which serves as a crucial branch point compound for the synthesis of aromatic amino acids and other essential metabolites such as the vitamin K or the vitamin B9. While I have focused on the overall roles of these key enzymes, the detailed catalytic interactions occur at the enzyme active site will be explained by Adam in the next section.
We'll now go over some key amino acids and cofactors necessary in each enzyme. We start with DAHP synthase, which consists of 4 active sites, each with a manganese 2+ cofactor necessary for enhancing the nucleophilicity of water to facilitate the reaction between PEP and EP4, with the other amino acids such as arginine, alanine, and lysine, which are involved in transition state stabilization. Then we have 3-dehydroquinate synthase, with 2 active sites and a zinc 2+ cofactor necessary for proton shuffling with histidine and the reagent and in facilitating the oxidation of a carbonyl to an alcohol. The phosphate removal is promoted by lysine, asparagine, and histidine, then followed by the aldol condensation by lysine, asparagine, and arginine. Dehydroquinate dehydratase can have either 12 to 6 active sites depending on the organism this enzyme is found in, with a proton transfer from the reagent to histidine 143 and a shift-based reaction of histidine 141. Shikimic dehydrogenase has 1 active site situated between the 2 globular structures of the enzyme, as seen above, with the NADPH binding into a Rossmann fold binding strongly with arginine and asparagine. The formation of shikimic acid is facilitated by a lysine and aspartic acid residue, with the lysine transferring a hydrogen to the reagent while the aspartic acid stabilizes the charge of lysine. Shikimic kinase has 1 active site that utilizes a magnesium 2+ cofactor to orientate both the shikimic acid and ATP and decrease the atomic distance between each of them to facilitate the phosphorylation reaction. EPSP synthase has 1 active site where lysine 22 deprotonates S3P, increasing its nucleophilicity to enhance its reactivity to PEP. Arginine 341 also transfers hydrogen, this time to the intermediate of S3P and PEP before deprotonating it once more to produce the product at the end of the mechanism. Charismate synthase is either a dimer or tetramer, having 2 or 4 active sites. FMN, or flavomononucleotide, is a cofactor in its reduced form to donate an electron to EPSP to promote the removal of a phosphate. Histidine 106 is responsible for protonating the reduced FMN to promote this electron donation. There is also histidine 17, which protonates the phosphate after the CO bond of the phosphate in EPSP is broken. This stabilizes the charge of the phosphate and forms the final charismate structure. And that is all for the key factors within each enzyme for the mechanism. I'll now hand it over to Noor Afizah for the applications of the shikimic acid pathway.
Now, I'm Noor Afizah, who will be explaining the application of the Shikimic acid pathway, focusing on its importance in medicine, food, and biotechnology. First, let's look at medicinal application. The Shikimic acid pathway is found in plants and microorganisms, but it is absent in humans, which makes its product very useful in pharmaceutical applications. One important compound is PABA, or para-amino benzoic acid. PABA is derived from charismate and functions as a UV absorber, helping to protect the skin from harmful UV radiation. Because of this property, it is widely used in sunscreen formulation and dermatological products. Another key compound is caffeic acid. Caffeic acid is derived from phenylalanine and is commonly found in coffee. It acts as an antioxidant and anti-inflammatory agent, and it has also been studied for its anti-cancer property. Next, we move on to food application. The Shikimic acid pathway also helps generate compounds that enhance stability and functional properties of food and beverage. Examples include gallic acid, caffeic acid, ferulic acid, and protocatoic acid. Ferulic acid is commonly found in cereal bran, wheat, and rice. It is added to nutraceutical and helps protect color and flavor during food processing. Another compound is protocatoic acid. It is often added to vegetable oils to prevent oxidation, used in fruit preservatives, and helps maintain flavor stability in beverage and canned fruit. Finally, let's discuss biotechnology. The Shikimic pathway is especially important in agricultural biotechnology, particularly in relation to glyphosate herbicide. Under normal conditions, the enzyme EPSP synthase converts EPSP into chorismate, which is the branch point for the synthesis of aromatic amino acid. This allows plants to grow and survive. However, glyphosate binds to EPSP synthase, preventing the enzyme from functioning. As a result, chorismate is not produced, aromatic amino acids are not formed, and the plant eventually dies. To overcome this, scientists engineer glyphosate-resistant crops by introducing a modified EPSP synthase that does not bind to glyphosate. This allows the plant to continue producing essential amino acid even in the presence of the herbicide. Finally, I'm going to be explaining the bioassay of the Shikimic acid pathway. The bioassay of the Shikimic acid pathway is the vanillin hydrochloric acid colorimetric bioassay, which is used to detect shikimic acid. Shikimic acid reacts with vanillin in the presence of hydrochloric acid, forming a pink-red colored complex. The intensity of this color is proportional to the concentration of shikimic acid and can be measured spectrophotometrically. This assay is highly specific for shikimic acid, making it useful for plant metabolism studies, especially in the shikimic pathway. It is also applied to assess glyphosate exposure, since glyphosate inhibits the pathway and causes shikimic acid accumulation. Overall, the vanillin hydrochloric acid provides a simple, sensitive, and reliable method for monitoring shikimic acid level in biological samples.
In conclusion, the Shikimic pathway is an important biosynthetic route that links primary metabolism to the production of aromatic amino acids. Through a series of enzyme-catalyzed reactions, simple metabolic intermediates are converted into chorismate, which serve as a precursor for phenylalanine, tyrosine, and tryptophan. This pathway is biologically significant because it is present in animals and microorganisms but absent in animals, making it a selective target for herbicide, antibiotics, and pharmaceutical application. Overall, understanding the shikimic acid pathway provides insight into how biosynthetic pathways function and why they are important in biology, medicine, and biotechnology. Thank you.